class was capped at 140 and there was supposed to be 140 seats. So sorry, we'll work that out. All right, so we are going to hit the high points of the syllabus. We are going to start talking through our chapter 1 content today. All right. So I posted the slides. They are just an outline, and I expect you to be taking notes during class. However that works for you is good. In the past, a lot of students have printed the slides out and written in as they go. If you can take notes on your laptop or whatever, that's great. My office is on the 3rd floor of Henry. It's right down the hall from the labs. I assume a lot of you are in lab. I know that some of you are not, and that's fine. I won't be testing you over lab material. Your lab professors, whatever, don't test you over lecture. So they're separate courses, so that works good. Right now, my office hours, I have them mostly in person. Fridays are going to be virtually by Zoom, so look for an email that will have that information on it. You're welcome to call me during my office hours. I usually don't, my office phone typically doesn't get much help or use, but you're welcome to call in. I may end up changing those virtual. We'll have to see how the semester goes here. But don't be afraid to ask if you want to meet with me another time. I'm around. I am teaching a couple of labs, but other than that, I'm around and available to meet with you. Okay, here's my happy family picture here. This was taken this summer. We had a chance to go up north for a day. I try not to talk about my family too much. I do make fun of my son occasionally because he has some kind of weird ideas and has asked some strange questions over the years, but this is my son. Whoops, let's see, gotta get my pencil working here. Okay, so here's our youngest. He's in his 3rd year at Michigan. He's an engineer, an engineering student. This is, I should start with my husband, Dr. Leonard. He's in the chemistry department, so some of you may have run into him. You can call me Professor Leonard. I actually don't have my doctorate. I do answer to Dr. Leonard, but I'm not actually a doctor, so just call me Professor Leonard. He's Dr. Leonard. This is our middle child. She has autism and goes to school in Hudsonville. Really excited to be back at school. And this is our oldest. She just started medical school, and it's been a long journey for her. So if you ever want to discuss that process with me, I'd be glad to do it. She had a couple of gap years, but now she's actually going to med school through the Navy and actually getting paid quite well to do that. So she's in Washington, D.C. At a school called UC's. So that's a little bit about me. I've been teaching this class enough that I'm starting to lose count. I think it's 7 or 8 times. So, and I've taught lab for about 15 years. So I really like microbiology. I usually do a little spiel to try to get students interested in microbiology, but it's so applicable now to decisions that you're making every day. So to say that it doesn't have anything to do with my life, you can't argue that anymore. In fact, you're probably sick of hearing about microbes. I'd probably like to hear less about them. But hopefully, once you've had this class, you're a little bit better informed. You can make better decisions about lots of things, whether to get vaccinated, how to clean a surface, lots of things to keep yourself safe. All right, let's see. Textbook. I've had a couple of you email me having trouble getting in with the code or something's not working. I actually got locked out of the website this morning, and I'm in the process of getting that resolved, so you're not alone. But, and a couple of you I've said, maybe see me after class and we'll see if we can work it out. This is the code that you will need once you purchase the, you go into the code. This is the course number that you'll need. So I've got that on the syllabus. Got it here again. That code should get you the e-text and access to the homework. All right, if it's not giving you one of those two things, then something's gone wrong. Don't just buy another one. Okay, we'll get it to work. I really like the textbook. I know it's expensive, but it's not too hard. It's not too easy. I'm not sure. I think that at some point they were going to stop selling the physical book. If you want that, this is the 6th edition. The 5th edition is really close to this. If you can get that cheaper, that will work great. Even the 4th edition is not too far off. So it's a good book. The nice thing about the e-text is it has animations right there in with the text that oftentimes are helpful. So again, I think it's a good textbook. And as far as technical requirements, we will be going online after Thanksgiving. So the final I'll be giving by Respondus lockdown browser. So essentially that means you can't use a Chromebook or a tablet or a phone to take that exam. So just kind of be aware of that. I assume that you're going to have to use that in a lot of classes, but we will be having some kind of little practice before we actually have to use it. Just to make sure everything's gone smoothly. I haven't given an exam yet that way, so it'll be new for me. All right, grading, the exams come kind of fast and furious. So about every 2.5 to 3 weeks, we're gonna be having an exam. You know, that's, in some ways that's good. If you know you don't do so well on one, then you have lots of other scores to bring it up. The final is worth a little bit more. And then you have this homework component that's close to about 10% of your grade. So it's going to give you a little bit of a bump. So if you got an A- and you're really close to an A, that might bump you up a little bit. It's not going to give you a whole letter grade. But I have this structured so that basically if you're completing your homework at about 80% accuracy, so for example, you've got about approximately 516 homework points. As long as you get 413 points, that's 80%, then you're gonna get the full 60 points of homework. Okay, I used to have it set up so whatever homework point you got, that was what points you got. So if you got, you know, 95% of your homework, you got 95% of your homework points. But I found that students were spending hours and hours and hours doing their homework to get every little thing right. And that's not really the point of the homework in my mind. The homework is just to give you some more exposure to the material, give you some practice. There's some case study type questions that hopefully will make you a little bit more interested. Again, it's just practice. If you come to a hard question and you don't know the answer, you might want to just guess every once in a while. But oftentimes you're gonna, again, want to put enough time into it to get about 80%. And most of you should get your full homework points with a little bit of effort. You probably don't want to save your homework till, you know, an hour before it's due, and then you're not going to do that well. But again, that's just a little bit of bump. I don't give any extra credit. The homework is your cushion, basically. Okay, and there's more questions about your homework if you look at the syllabus a little bit more in depth. Okay, so students always ask me, well, how do I do good in this class? You may have heard horror stories about this class just because it's got a lot of content in it. So we do expect, even though you're only in class about 3 hours a week, well, less than that, you're going to be spending double or triple that amount of time outside of class doing readings and going over your notes and doing homework. So if you're working 20, 30, 40 hours a week, it's going to be really hard to do well in this class. So think about your priorities there. If possible, you might want to read the text before lecture. I know that this is not probably your only class, and in a perfect world, we would do that. Probably more often, you're going to use the textbook as a reference. So a reference for when you're doing your homework. Maybe we do something in class that doesn't make sense to you the first time. go back and read about it in your textbook, that section, and hopefully it will make a little bit more sense. There are a few chapters later on where I'm going to really encourage you to read the assigned reading. That's really going to help you get a full picture. But for the most part, it's okay to kind of use it as a reference, I think. The PowerPoints that I posted, like I said, they're just an outline. I'm going to be expecting that you take some notes during class. I have changed the way that I lecture this semester. Hopefully there's not too many bumps along the road, but my daughter made the comment to me this summer, something about, she used the term death by PowerPoint. And wait, I love PowerPoint. What do you mean? I understand that just, you know, having somebody read PowerPoints, it gets a little bit boring. So I'm trying to make you a little bit more involved by writing in some content yourself. Okay. But it is a great way to get across a lot of information efficiently. And I do only like to test you over things that we've done in class. Okay, I'm not going to be testing you over things that were in your reading assignment. Okay. I would encourage you to do that homework, that online homework, within a few days of after we cover it in class. I always have a few students that wait and do all of it, you know, the night before the exam, and then you've got maybe 4 hours of homework to do, and that's not a great way to study. So I would do that pretty soon after we do it in lecture. So after today, you can take a look at that chapter 1 homework. I do have your first homework assignment due a week from today, and that's just a 2-question getting started in mastering. So that's just to try to encourage you, get that textbook purchased and make sure you're not having trouble log in. I can extend that if you're having some problems, you know, getting your textbook or getting logged in. But again, that's just 2 questions, it's not content. Your homework, typically I have set as being due the day of your exams. Also, I just want to encourage you to learn the terminology. So with any biology course you have, there's going to be a lot of terminology to get through to kind of understand what's going on. If you're just reading through those words without knowing what they mean, then it's just going to snowball and you're not going to understand. So if you come to a term that you don't know, take the time to figure out what it is. I typically have my final exam structured with a whole section on matching of terms. So again, that's usually the easiest section of the exam because you have things as different as an electron microscope and a phagocyte. Like, you know, it's pretty, you know, not detail oriented, main idea type things. All right, so just a little perusal through what you're going to be studying this semester. This course has a very clinical bent to it. So a lot of introductory microbiology classes might give a little bit more service to like a survey of microbes and talk about ones that live in the environment as much as the ones that live in our body. But we don't really mention much soil organisms or waterborne microbes. We concentrate on the ones that are in our bodies because a lot of you are probably here because you're going to be doing something in the medical field. So that is a little deficiency in our microbiology major right now, that we don't have an environmental microbe course in BMS. There is one in biology, but that course is designed, it has a lot of overlap with this class, unfortunately, so we don't really encourage you to take both of them because they're covering a lot of the same thing. Okay, so we're starting out with just basics of mostly we're talking bacteria, but we do talk about other microbes as well. A lot of content, a lot of new stuff you may have had quick exposure to in other classes, but this, I assuming, will be mostly new. After that first exam, it gets a little bit more conceptually difficult because we're thinking about metabolism and genetics of bacteria. There should be things that you've seen before here. So you've probably talked about metabolism from a human standpoint before. We're going to emphasize things that bacteria can do that are different than humans, differences in transcription, translation, replication in bacteria from humans. So, but that should look a little bit familiar, but again, it is a little conceptually more difficult. Students tend to find that a little bit more difficult. And then the exam 3 material typically gets a little easier. Again, it's a little bit more cut and dry stuff, but now we're starting to get real application. How do we kill microbes, both in our environment and in our bodies by using antibiotics? So let me update this fall break. Sorry, we've lost that. That does give us a little bit more time to talk about antibiotics on the bright side there. When then we're talking about the process of infection. So again, really applicable. This drop deadline, this has been changed as well, and have that on your syllabus, but just didn't update this one. So a later drop deadline to give you a little more chance to decide there. And then at the end of the course, we get a little bit more conceptually difficult. Again, we're talking about how our body fights off an infection. So a whole section on immunology. That's the section that I would encourage you to read your book as well, because it is quite complex. The last couple weeks of class, which we will be, oh, and I wanted to mention, just give you the heads up, this exam, November 24th, that's the day, that's a Tuesday before Thanksgiving break. So I know that will be painful to take a big exam right before you leave for break, but I certainly didn't want to do it after a break, and that's just sort of the way it works out. If you know that that might be a problem, make sure you talk to me and I'll maybe get you to take it earlier, but that's why I had to do it. After break, we will be online, so I'll be giving recording lectures and posting those. We pick out certain particular infections and then think about the things we've talked about all semester that are specific to that infection. So tuberculosis, for example, what's special about that bacteria that enables it to cause an infection? What's special about its structure? How do we treat it? Is there a vaccine? So that's really applicable, and students tend to like that portion. We call that the bug parade portion of the course. Okay, any questions about the syllabus? Again, take a look at that. I know it's a long document, but take a look at that. Okay. All right, so I try to start out, whenever we start a new chapter of material, I try to throw up some questions, just kind of, hey, this is where we're going today. One thing I forgot to mention, I must have skipped over that somewhere. I do have learning objectives posted. So there's a tab right there on Blackboard. It says learning objectives. It's like 17 pages long. I would encourage you to go ahead and print that out here at some point. Then you have those learning objectives. That's what I look at when I'm making an exam. And I try to make sure my questions fall under one of those learning objectives. So, you know, some students write out answers to all those learning objectives, and that's a lot of writing. You can do that if you're one of those students that are really good at keeping up with things, but those are there for you. That's another tool. Some of them I've written myself. Some of them come straight from the textbook, and then I have it in parentheses where you would find it in the textbook. All right, so these questions that I'm posting, I know not every slide that I show here are ones that I posted. If I show you slides that I didn't post, that's because I decided that it wasn't really something that you would need to study from, basically. So this is where we're going today. A little introduction on what microbiologists study, very brief on how we classify them. We're gonna get more into that next time. From time to time, I like to look a little bit more in depth at famous experiments. So we're gonna do that with the spontaneous generation. And what causes fermentation? That was another early problem that microbiologists worked on. What causes disease, and what can we do to prevent disease? So I'm focusing on kind of famous people that have helped us to solve these problems. Okay, so you notice, if you compare my lecture to the text, I touch on a lot of things in the text, but there's certainly things in the textbook that we won't get to in class. All right, so first of all, when did we first see microbes? Antoni van Leeuwenhoek, he is who we credit with making the first microscope, and he was a Dutch cloth merchant. Okay. And so this was a picture of his early microscope. And you would never guess that it was a microscope unless you knew that. This little part right here, this is actually the lens of the microscope. And he was really good at making high quality lenses. And to me, when I remember first learning about Leeuwenhoek and thinking he was a cloth merchant and he made lenses. That seems like a really odd combination to me. But he was Dutch, and think about what, I don't want to say biases. When you think of a Dutch person, you have certain things that come to your mind that they're tall, okay, right? Dutch people are tall, they're blondes. What else? They like pea soup, right? They're cheap. No offense to anyone, but Dutch people are known for being thrifty. They're also very clean. That was another thing. But he wanted to make sure that nobody was cheating him on him. When he bought some fine quality cloth, he wanted to be able to inspect that really closely to make sure it actually was fine quality cloth. So yeah, if you go Dutch on a date, that means, you know, the guy maybe is a little cheap, right? All right, so he got really good at making these lenses. This little pointy part right here is where he would, you know, stick whatever he was trying to look at, and then he would hold it up and look through that little lens at whatever the specimen was. So he not only looked at cloth, he looked at all sorts of different things, drops of water and organisms, and you name it, he looked at it. And he wrote up his results and published them, and he was actually a very noted scientist of his time. But he never published how he made his lenses. So in terms of making microscopes, that science didn't really get, you know, better for a long time. But if we think about what sort of little organisms, he called them wee animalcules. If we think about what sort of organisms he saw under his microscope, we could classify them in 6 different categories. Okay. He saw bacteria, archaea, and of course, he didn't classify them as that back then. More about that later. Fungi, protozoa, 2, 3, 4, missing. Algae, and we'll say small multicellular animals. Of course, what he couldn't see, and what we as microbiologists study today, we couldn't see viruses, but I'll put that in parentheses, right? We weren't able to see viruses until we invented the electron microscope, which was in the 1930s. Right? So he saw all sorts of things. Carolus Linnaeus is another important name to be familiar with him. He's known as the father of modern taxonomy. Okay, and he lived, you don't really need to know years here, but his life overlapped with Leeuwenhoek's. He lived in the 1700s. I wouldn't necessarily call that modern, but okay, whatever. He's credited with really organizing the scientific community around classifying organisms and naming them. So his system, he had plants and animals. Okay, so that's where we started, and we've built on that and been changing that classification system ever since. So we're going to talk about him a little bit more next time when we get into a little bit more depth of taxonomy. Okay, so if we do a little survey of those kinds of organisms that Leeuwenhoek saw, these are the kinds of things that microbiologists study. Okay, prokaryotic organisms, they're ones that, right, this should be reviewed, they don't have a membrane surrounding their nucleus. Okay, and there's two kinds of prokaryotes that we classify today, the bacteria and archaea. We'll talk more later about how you know it's one and not the other. You can't necessarily tell just by looking at them. They're both very, very small. The archaea are typically known for being a little bit more extreme microbes, are living in extreme environments. Not always the case, though. We now know that there are archaea that are part of our normal microbiota. But here's an example of an archaea down here, methanogens. They're organisms that actually produce methane as a byproduct of their metabolism. But there's archaea that live in the hot springs of Yellowstone and, you know, the polar ice caps and all kinds of strange environments. The bacteria are mostly what we're going to be focusing on in this class. Here's an example of Staphylococcus aureus. It's an organism that we'll talk about a lot and you will be using in lab quite a bit if you're in lab. Here's another little more bizarre example of a bacteria. It's called an organism called Deinococcus that actually lives quite happily in radioactive waste. Okay, it has special protections for its DNA so it doesn't get mutated. All right, so we'll hopefully you'll understand as we go on that really there's life that lives almost in any kind of niche on the planet. So prokaryotes living everywhere. We are going to spend a little bit of time, much less, on eukaryotes that are microscopic. There are microscopic fungi. This is the mold Penicillium chrysogenum. So that's the mold that actually secretes penicillin for us. Yeast is a microscopic fungi as well. Protozoa, we don't spend a lot of time on them, but there are a few very notable disease-causing protozoa. We often classify them based on how they move around, their locomotion, so whether they have pseudopods or cilia or flagella, but we'll talk about them kind of briefly a little bit later on. Algae we don't talk about much at all, even though they're really important for the Earth's food chains and they provide a lot of oxygen. They're not disease-causing, so I don't really worry about them too much, but here's an example of a freshwater algae, Spirogyra, and these are diatoms, marine algae. And there actually is a difference between algae and plants that we'll talk about a little bit later as well. I didn't post this slide about parasitic worms. Some of them do have microscopic stages of their life cycles, but we don't really get into them at all in this class. We do have a parasitology class that Dr. Graham teaches, and it's really interesting because he's had a lot of these infections himself, but so take parasitology if you're more interested in that. Okay, and lastly, I'll mention viruses, of course. Oh, there it is. This actually is the SARS virus here, the first coronavirus, as an example there of a virus that infects animals, humans, and here are some viruses that are infecting a bacterial cell. We call them bacteriophage. Okay, but just briefly, what are viruses? Basically, they are composed of some nucleic acid. So they might be DNA or RNA, not both, one or the other. And that's going to be covered in a layer of protein. So they're going to have protein coat around them. Okay, so here is the genetic material, the RNA and the SARS virus, and then these little bluish, greenish proteins that are protecting it. Some viruses also have a membrane as well. All right, so more about viruses a little bit later. Okay, whether they're alive or not, we'll get into that debate as well. Most scientists would say no. Okay, so that's a little bit about what microbiologists study. Like I said, from time to time, I think it's important to think about and learn about how scientists think. Okay, as educators, we're always being told you can't just be cramming content down into students' brains. You have to teach them how to think, right? So how do you do that? Well, you learn about how historically we have solved problems. So if you look at this very famous set of experiments, how did we begin to understand that life has to come from life? That you can't just throw some corn into a piece of flannel shirt in the corner of the barn and a few weeks later have mice there. Okay, baby mice come from bigger mice. All right, so the story begins with Francesco Redi. He was an Italian physician. And I just added this year in there. Again, don't learn years. I always had trouble bringing it to mind, so I added it in. But mid-1600s, so a contemporary, he overlapped with Leeuwenhoek a little bit. Okay, so he set up this experiment to try to prove that life had to come from life. All right, so he had an unfilled flask, a flask that was open to the air, that he put some raw meat in. He had another flask that he kept closed. So that was kind of like his negative control. He didn't expect life to be there, okay? Or baby flies is what he was looking for. And then he had a third flask that he kept open to the air, but it was covered with gauze. So flies couldn't get to that meat which they were attracted to. Okay, so he found that there was baby flies or maggots on the exposed meat after waiting a few days. No maggots here on the sealed flask, and the flask with gauze actually found some maggots on that gauze, again because the flies were attracted to that scent. Okay, so this was a really good first step. This, you know, most people, I don't know how well science was disseminated in those days, but at least scientists were believing that life had to come from life. So this helped to disprove abiogenesis. But about 80 years later, somebody did an experiment that kind of muddied the waters a little bit. Okay, John Needham, he published this work in 1748. And at that time, you know, we were starting to learn about these wee animalcules, these microbes that Leeuwenhoek saw. And we understood that, you know, multicellular life came from life, but what about these microbes? Could they just spontaneously generate? So he took a flask that he had chicken broth in there. So a source of nutrients, and he heated it up and sealed the top, let it sit for a few days. And so after a few days though, the broth started to get kind of cloudy, okay? And we know that once suspension starts getting cloudy, they get cloudy because there's microbes growing in it, okay? So he came to the conclusion that microbes could come basically from nothing. They didn't have to come from other microbes. And we didn't have peer-reviewed journals in those days. This was hailed as a you know, great experiment, and he was elected to the Royal Society of London and all these things. About, let's see, I think this was 1799. Lazzaro Spallanzani, he was an Italian priest, actually, So about 50 years later here, he did a very similar experiment, because maybe he didn't believe Needham's experiment. Basically, he took some gravy, again, kind of a slew of nutrients there, and he boiled it in order to kill any life that might be in it. With one flask, he stoppered it or sealed it, left the other one open, and predictably, the one that was open after a few days was teeming with microorganisms, and the other flask was free of microorganisms. All right, so what did he conclude from these experiments? He concluded that Needham didn't boil his suspension enough to kill everything in it. So Needham failed to heat the vials sufficiently. And that microbes exist in the air and can contaminate experiments. So from this, he concluded that even microbes cannot spontaneously generate. Okay, so again, probably Needham didn't boil his flask long enough. Kinds of microbes that are really hard to kill, that you might have to boil for much longer. Okay, but still there was people that argued, well, he's not letting air in here, and perhaps those microbes need air in order to spontaneously generate. So there's one more experiment that Louis Pasteur did that really kind of settled the question once and for all. And Louis Pasteur, he had a little bit of an ego. He even said, never will spontaneous generation recover from the mortal blow of this simple experiment. Okay, so he designed this really well-designed experiment. He actually knew how to blow glass, and he made these special swan neck flasks and put his gravy infusion into that flask. And the great thing about these flasks is that they were open to the air here. So he boiled that infusion for a long time and then just let it sit, and indefinitely no microbes. That solution didn't get cloudy at all. So basically any microbes that would float in from the air would kind of get stuck in that U-bend there instead of floating up. All right, so again, this was kind of how the process of how that whole problem was solved. And Louis Pasteur is famous for a number of other things. Looks like from time to time my writing gets a little squished up there. I know that's kind of small for you to see on the big screen, but Pasteur is famous for a lot of other things. He made the first anthrax vaccine, the first rabies vaccine. He also did, basically he's credited with saving the wine industry in France. So at that time, they didn't really understand the process of how grape juice came to become wine. They called that process fermentation, but they didn't understand how that happened. And sometimes the wine would turn out bad. It would be bitter, and they didn't understand why that happened. So this is a good picture of how science is supposed to work. You look at basically the scientific process. So Pasteur made the observation that grape juice fermented into wine, and when he looked at that wine under a microscope, he could see bacteria and yeast. Okay. So after he made that observation, he made some hypotheses and designed experiments to prove or support or disprove those hypotheses. So his first experiment was spontaneous fermentation just happens. Basically, grape juice just becomes wine. So he made a flask where he killed all the life in the grape juice there, and then over time, no fermentation, right? So you can reject hypothesis one. Didn't happen on its own. What about, is it the air? So he allowed air in his flask, but still no fermentation. So you can reject that hypothesis. What about bacteria? Is it bacteria that do the fermenting? Well, he basically only put bacteria in that grape juice. He killed everything else in here. But he found that the end products of bacterial fermentation are usually acidic. So that made a very bitter, bad tasting solution. So he rejected that hypothesis as well. And then if you think, well, maybe it's the yeast then that does the fermenting, and he tested that and was able to accept that hypothesis that when you make sure there's no bacteria in that grape juice, and then you add the yeast, you get good tasting wine. So he developed a process that we call pasteurization after him today, where you heat up that grape juice enough to kill all the bacteria, and you add the yeast, and then you've got the good tasting wine. So a lot of times today, with the idea of making money, you'll see science not working the way that it was intended to. So companies have something that they want to prove in order to make money. So they start with some conclusion and try to work back the other way. Try to design experiments to prove, say, for example, that milk is really good for you. The milk industry is really suffering for a while. If they can get more people to drink milk that it's good for them, then they'll sell more milk. So sometimes we work back the other way, and then that's just not good science. Okay. All right, so thinking about other problems, that era from the 1870s to about the 1920s was really what we call the golden era of microbiology. It's when Pasteur was doing his work, and that work he did with fermentation and disproving spontaneous generation led to what we call the germ theory, where we understood that it's germs that cause a lot of disease and not just bad air or miasmas. So Pasteur contributed to that work as well. Again, we call that the germ theory. But another really famous contemporary of Louis Pasteur was Robert Koch. Pasteur was French, Koch was German, and they hated each other. They were on opposite sides during the Crimean War, and there was probably some misunderstandings caused by translation of some of their correspondence, but they hated each other, and they worked on very similar problems. Koch is best known for trying to understand the etiology of disease. Okay, and that just means the causation of disease. So he was known for being a really meticulous experimenter. He was the first to elucidate what caused anthrax. It was a bacterium called Bacillus anthracis. Okay, and here's a picture of Robert Koch. All right, so he is best known for, he also figured out the causation of tuberculosis. Before that, we thought it was a cancer because that infection is really slow acting, but he proved that it was another bacteria. And he used something, a process that he referred to as Koch's postulates. Okay, and we still, to some extent, kind of follow these postulates whenever we're trying to understand what causes some infection. And they're not perfect, you can't use it in a lot of different cases, but this is a good early step. So this graphic is not from your book. Your book doesn't have a nice picture to go with it, but I think it kind of helps us understand the 4 steps that you need to satisfy in order to prove that a particular organism causes a disease. Okay, so postulate 1, the suspected germ has to be present in every case of the disease. So Typically, you're working with a lab animal, and you have maybe these lab animals getting sick, and you're able to isolate a particular organism. So you have to be able to maybe, take some kind of a tissue sample or a blood sample or whatever, and streak that out on some microbiological media. We're going to talk more about media, probably getting into next time, a little bit today maybe. And you have to get that particular germ all by itself. We call that putting it into pure culture. So you've got that one organism, and you take that one organism and you inoculate it back into your test, a healthy mouse, for example. And if that mouse gets sick the same way as before, and you can isolate that same germ from it, that's pretty good evidence that that germ is what's causing the illness. And I should say also that typically you can't find that germ in any mouse that's healthy and not ill. Okay, so this we began very quickly to understand the causation of lots of infections using Koch's postulates. And there are lots of instances where this is not going to work, right? For example, if you have a viral infection, you have to, it's a lot more difficult to isolate the virus and grow it up. You have to use different culture methods to grow it up. Some infections are found in healthy people as well as sick people, right? We see that today. A bunch of you are quarantined that aren't here today because you're not showing symptoms, but you may have the COVID anyway. There are some infections that there's lots of different organisms that cause those infections, like pneumonia. So this is not a perfect system, but again, it was a good first step. So in order to satisfy Koch's postulates, we had to get better at seeing organisms, viewing them under the microscope. Hans Graham, I believe he was in Robert Koch's lab. He developed the Graham stain, again, about that same time. And if you're in lab, you're going to get to do this quite a number of times. And we'll be talking about probably next week what the difference is between a bacteria cell that stains gram positive and a bacterial cell that stains gram negative. Differences in their cell wall. But that's usually the first question we ask when we're trying to identify a bacteria. Is it gram positive or gram negative? Two big groups. Okay, there are always exceptions and organisms that don't belong in one or the other, but that's usually the big first question we ask. I want to mention a few people that are well known for making great advances in preventing infection and disease because they contributed to us understanding that hygiene is really, really important. Ignaz Semmelweis, in 1848, he was a Hungarian physician, and he advocated hand washing in his hospital for physicians. Okay, so he noticed that physicians were going straight from doing autopsies to delivering babies, and they often wouldn't even wash their hands. And so he didn't really understand what it was about hand washing that might make a difference, but he made good statistics and saw the death rate from women dying of childbed fever went from like 12% way down to like 1 to 2%. So he proved that this was making a difference, but because he didn't understand, this was kind of before we understood the whole germ theory thing, we didn't understand what caused infections. Other doctors were offended that they might be causing women to die, and he was kind of laughed out of his profession and ended up dying in an insane asylum. But he did publish his work, others took note of it, and Lister in the 1880s, he was an English physician, and he went a step further from handwriting. Now the whole germ theory thing was a little bit more accepted, that germs cause disease, and he used carbolic acid, which is actually phenol, both in the operating room and in hand washing. So he actually sprayed it as a mist in the operating room. He washed wounds with it, or surgical incisions. Physicians washed their hands in it. And this drastically cut the infection rate during operations. Unfortunately, if you're wiping phenol on your hands every day or breathing it in, that's not really so good for the physician's health. So it caused their hair to fall out and their hands to get bleached and their fingernails to fall out. But it did cut infection. Okay. And then Florence Nightingale, she was a nurse during the Crimean War and documented a decrease in infections due to better hygiene. So basically cleaning hospital wards and dressing wounds, and she just documented how much improved outcomes she had, less infection, and so she's famous for that. Just a couple more people. John Snow, again, we're kind of going back a few years here. He's known as being the father of epidemiology. Okay. He traced a cholera epidemic in London, and to try to figure out what was causing it. So at that time, we didn't understand cholera is a bacterium that is in the water supply. So right back then, they didn't treat their water supply. And he went from house to house in London and asked people, do you have anyone ill with cholera? And he made very, comprehensive notes about this, and made a map like this one. The red spots were households where somebody was ill with cholera. And he noticed a big cluster of cases around this little blue dot here. The blue dots here are the water pumps. So he took the handle, this was the Broad Street water pump. He took the handle off the pump and the cholera epidemic subsided. So they found that the source of contamination there was in the water by that pump. So anytime you want to find a simple solution to a complex problem, you say, where's the handle to the Broad Street pump? Okay, and in terms of preventing infection and treating infection, a couple names you should know. Edward Jenner, he's responsible for, or we give him credit for having the first vaccination. Okay, let's see. Smallpox was a constant scourge, deadly scourge, and he noticed that milkmaids that had had cowpox very rarely, if never, got smallpox. So cowpox was just kind of a rash. It was very mild. And he took some scabs from a milkmaid that had cowpox and used a knife to kind of scratch it into the skin of a boy. And then he intentionally inoculated the boy with some smallpox lesions, and he didn't get sick. So not quite the way we do our clinical trials today for vaccines, but it worked. And it was much safer than actually inoculating someone with actual smallpox scabs and trying to get a mild infection, because people died from that. All right, and then Paul Ehrlich, he was a German scientist. We credit with really being the first one to try to come up with drugs to treat infections. So he was searching for a magic bullet. Okay, so a bullet that's not magic is going to kill anyone, right? But a magic bullet would only harm the bad guy and not harm the good guy. So he was looking for compounds that would search out and kill a particular germ. In this case, he was looking for compounds that would cure people of syphilis. It's a bacterium. So he wanted a compound that would, again, kill the syphilis and not harm the host. And he found on one, happened on one he named salvarsan. It was an arsenic compound that helped rid people of syphilis. Unfortunately, anything with arsenic in it is going to be pretty toxic to humans too. So it was not a great treatment, but it was sort of the first step forward. Okay, so that's pretty much what I have for you in terms of chapter 1. Okay, so it's pretty clear in the objectives which people I want you to just kind of know what they're famous for. Any questions here as we move forward? We have a little bit more time to get into chapter 6. Okay. All right, so we do jump around here a little bit. If you've seen the syllabus, I'm bouncing around to a lot of different chapters. We typically do that at the beginning of the semester because we're trying to match up with those of you that are in lab, what you're doing in lab. So we're gonna just bounce in chapter 6 kind of briefly here and talk about culturing bacteria and a little bit about microbiological media. And then we kind of bounce over to chapter 4 and talk about microscopy for a little bit. But we're just doing a little piece of chapter 6 and then we're going to come back to the rest of it later on. So you'll see that your chapter 6 homework assignments, there's 2 of them. The first one is just this little bit about culturing media, and then later on we'll get more about growth and counting bacteria. Okay, so first, just thinking about why do we want to grow bacteria in the lab? Well, we need to be able to do that in order to accomplish Koch's postulates. All right, so we're gonna isolate an organism. We have to be able to grow it up all by itself. We have to have a way to do that in lab. And that's not necessarily easy to do. How do we grow bacteria? So like I said, we're gonna talk about some medias and just a little terminology that we use when we're growing bacteria. Pure culture is a really important term. We want to keep organisms all by themselves because if we're trying to identify that organism and there's actually 2 of them there, it's going to give you all sorts of conflicting results. Okay, and then a little bit of terminology to do with different kinds of media, just to kind of understand some principles there. And a lot of this is overlap with lab, but that's okay because it's kind of important stuff. All right, so a little terminology that can be confusing. We can use that term culture as both a verb and a noun. So if we're using it as a verb, that means we're going to basically grow up. We're going to culture an organism. We're going to grow it up on some media. Okay. A culture, then, when you use it as a noun, is the place where the microorganisms are growing. Okay, so a culture can be right here, like in this picture here, it can be a petri dish with media in it. It can be a test tube with some liquid media in it where bacteria are growing. Okay, when we inoculate a culture, that means we're going to introduce an organism into a culture. Okay. And you can see here, this is kind of hard to see, but this person is using a wire loop to inoculate organisms. Well, that actually looks like in this picture, he's taking some organisms away. But if we were starting a new culture, we often use that wire loop to get some cells from one place and then mix it in to whatever sterile media you have. So that's inoculate. We use that same root as a noun, inoculum, and that refers to what organism is used. to start a culture. Okay, so in some cases we might talk about the inoculum quantity, so how much of the organism you use to start a culture, right? And then of course the medium is just the collection of nutrients where the organism grows. Okay, and we'll be talking about some different kinds of mediums here shortly. A word that it's easy to kind of confuse, inoculate kind of sounds like incubate, but of course they're very different things. We typically have to incubate our cells on that media in order to get them to grow. At least they're going to grow a lot faster, a lot of them, if you incubate them at their ideal temperature. So that just means, again, in order to get the cells to grow up, we need to put them at usually a nice warm place. The organisms that we use in lab, we usually incubate between 35 and 37 degrees Celsius. Of course, 37 degrees Celsius is body temperature. I think we have our big walk-in incubator set at 35. It's close enough. Okay, and typically the organisms that we use, anyone, again, that would live in our body, they typically grow up within a day or two when you put them at that nice warm temperature. And another term, so you can see here a loop that's being used to rub some cells on the surface of a medium. Okay, there are cells most likely being rubbed on there. Of course, you can't see them with your naked eye. You have to give them a chance to multiply. And once they grow up enough, then they grow into colonies. Okay, and the colonies then you can see with your naked eye. So there's probably millions of cells that make up a colony. Okay, and again, you'll get to see that not this week in lab and not next week because we don't have lab next week, but the week after. Okay. Let's see. Oh, and I should say, I think I'm going to talk about this in a couple more slides, but if you have a colony that's all by itself, like this one that's not touching any other ones, we call that an isolated colony. Okay. And that's important. We like to get isolated colonies because you know all the cells in that one colony, they're essentially clones of each other, started out as a few cells and they just kind of divide, so they're all the same thing. When you've got some colonies growing together like this, you could actually have a couple different kinds of organisms all growing together. Now, in this case, it's all that same pretty unique color and all the colonies look alike. So we like to be able to see those colonies off by themselves to make sure that it's kind of all the same thing. So I think I'll talk about that again in a second. All right, there's really two basic kinds of media that we use to culture organisms: liquid media and solid media. In liquid media, we tend to refer to as broths. Okay, and liquid media is really good for growing large quantities of bacteria. Okay, so the way you would make some liquid media, you would take, say you wanted to make some media called nutrient broth. You buy that in powdered form and you measure out the right amount, you put it in a flask, you add the appropriate amount of distilled water. Maybe you have to mix that up a little bit by using a stir bar and a magnetic plate. Mix it up, and then we sterilize it by using an autoclave. We'll talk more about that later. And then you've got your sterile media and you're ready to go. You could inoculate that with some bacteria, maybe in a big flask, and then overnight, it would look really cloudy. The more bacteria were growing in it, the more cloudy it would look. So again, here's that clear media. You can't always tell by looking at it or not whether there's cells in it or not. There could be a few cells, of course, and you can't see it. But the cloudier and cloudier that media gets, the more bacteria are growing in there. And sometimes there's enough that it settles out kind of a little pellet in the bottom. Okay, but that's one way we can kind of quantitate how many cells are in a tube, is by testing how cloudy it is actually. Okay. But one thing that liquid medias are really bad at is being able to tell if you've got one species of bacteria in there or 20. You can't tell just by looking at that. So back in late 1880s, we actually developed solid media for the first time, and Robert Koch is credited with being the first one to add a component to his liquid medias called agar. There was somebody in his lab that I think her picture is actually in your book. Her last name was Hess. She said, I use agar in my jams and jellies at home. Maybe we could add that to our broths in order to solidify them. We've been using it ever since. We haven't found anything better. So basically you might have some nutrient broth and you add agar to it. And now when that broth is still hot enough, or when that nutrient agar has been heated up and it's still hot enough, you can pour it into any shape that you want. You can pour it into a petri dish, and then you've got a plate. You can pour it in a test tube and allow it to cool at an angle, and we call those slants. Okay, so again, if you're in lab, you're going to be inoculating some plates and slants and broths, the first lab that we do. What is agar exactly? This is actually a polysaccharide that's actually found in the cell walls of a particular kind of seaweed. It's actually a red algae. Okay, and what's so great about agar is that most bacteria don't have the enzymes in order to break those bonds between the polysaccharides. So the bacteria cannot digest, or they cannot use the agar as a nutrition source. And that's what we want. Gelatin sometimes is used as a solidifying agent, but there's some bacteria that can break down gelatin. So when you inoculate your solid media and then you incubate it and let your bacteria grow, it turns to mush because the bacteria are breaking it down. Okay, so agar is, again, a solidifying agent. Okay, it's not a source of nutrients. We don't want the bacteria to actually eat it. That's what we want. Okay, and how do we get agar? It turns out we can't just farm this seaweed. All attempts to grow that and farm it have failed. We have to, I didn't post this, but we actually, after storms in certain areas of the world, like Morocco and Spain, South Korea, people go around and collect this certain kind of algae that washes up during storms, and that's where we get algae. This was from an article. They were concerned 4 or 5 years ago that we were over collecting it and that we were going to have shortages, but that didn't really seem to pan out. All right, so when we have solid media, it's a lot easier to get pure cultures. And a pure culture, that just means there's only one species of organism in that plate or in that test tube. Okay, this is typically what we're trying to do in lab. We like to work with one species at a time. We don't want other things contaminating our culture and kind of confusing our results. So we like to keep our cultures pure. And ways that we get a pure culture from a culture that's not pure, we would call that a mixed culture. one way that you're going to be doing in lab quite a lot is doing a streak plate. Okay, so that means when you put your cells under your solid media, you streak it in a very particular pattern in order to try to get isolated colonies. Because once you get these colonies isolated, you can see, oh, they all look the same. I'm pretty sure that this is a pure culture because all of those discrete colonies look exactly the same. There are a couple different ways to try to get isolated colonies. You can do a spread plate or a pour plate. We used to do both of those in lab, and I think probably we're not going to be getting to that this semester. So I just mentioned it briefly, but a spread plate, you use a little glass hockey stick after you put your bacterial suspension on a plate, and you use that hockey stick to just kind of spread it around. You can get isolated colonies that way. A pour plate, you actually add your bacteria into the agar when it's still hot enough to be liquid before it cools. So that's why you were sterilizing it. After that, you add your suspension in there. And then when you pour it into your Petri dish, you've got bacteria suspended throughout the agar and not just on top of it. Okay, all of those ways are going to give you isolated colonies. Again, the streak plate is probably the one that you're going to be most familiar with. And the most important thing here is that how do we keep those cultures pure? We do something called aseptic technique. And I'm going to let your lab professor talk more about that. I'm not going to really go into it in lecture here, but these are techniques that we use to try to keep our cultures pure. So things like keeping the lids on the plates so bacteria in the air doesn't contaminate it, or making sure our loop is sterile when we're transferring it, wearing a lab coat so spores don't shake off of your clothes. Okay. We will pick up there next time. I'll be posting some more notes today or tomorrow, so look for that. Thanks for coming, and have a good day.